Good afternoon, everybody. As it is the need of the hour, the university is organizing a global webinar for the information of our faculty and students, because now after the COVID, people are very much anxious and concerned about the re-emergence of or emergence of diseases. Of course, this particular disease, human metapneumovirus, has been there since the last 2020 to 2001 year, and now it may be a re-emergence or emergence. So people are very much concerned about it. Media especially is producing a lot of anxiety and panic. So our Honorable Vice Chancellor was inquired about it by the media, and that is how this webinar is being arranged. So I don't want to spend too much time between the experts and this person, my talk. So I request everybody to mute during the webinar unless one is not talking, and also please follow the etiquettes for such social media activities. And let me come to my duty. I wholeheartedly welcome our Honorable Vice Chancellor, whose inspiration and support only led to this program. And I also welcome our international expert, Dr. Shyam Kotilil, who is from Maryland University. I welcome our own in-house faculty, Dr. Suma Sri Kumar, who is the Executive Director of the Institute of Advanced Virology, Aravind, who is the Head of the Department of Infectious Disease from Government Medical College Trivandrum, and of course, our senior, who is the Associate Professor, School of Public Health. Now, I also welcome Dr. Sharada, who is the HOD of Microbiology. Formerly, she was the HOD Microbiology from Government Medical College Trivandrum, and now serving at SUT Medical College. Above all, I welcome all the participants who are eager to listen to these expert persons, and please use the chat box maximum for interactions. I hope Dr. Shyam is very busy, and Shyam.
Yeah, he has come. He's there.
Okay, very good. Because Shyam told us that Shyam need to leave because of the climate problem there, because it is -15 or -20 many times. So I request our Honorable Vice Chancellor to say a few words. Thank you, sir.
Good evening, my colleagues in India, and very good morning, Dr. Shyam Kotilil. In fact, day before yesterday, large number of media persons asked, what is happening? Are we going to be in a lockdown situation? And after COVID, something else. And again, this is reported from China. When somebody is reported from China, people are worried whether it is fully reported, what is it? So we thought day before yesterday, I've been in the School of Public Health, and we had a brief discussion with Dr. Rajmohan and Dr. Suma and other colleagues, and we thought it is better to have a good knowledge sharing, so that all of us will know, all of us will be able to tell everyone that this is the position. This is a bad situation, this is a good situation, nothing to be worried. And in between, I talked to Dr. Sri Kumar from IAV, and he said he has seen it last 2-3 years, the same virus infection has been seen. So we thought we will have Dr. Shyam Kotilil, who will give us the international perspective, and Dr. Sri Kumar, his experience with this infection, if at all it is here, and how common it is. And of course, we have our experts, Dr. Aravind. Aravind will be telling us what are the clinical problems we are likely to face and how do we tackle. Sharada Devi definitely will tell us about the virology part, and Dr. Suma will be acting as a moderator, and Senior from the School of Public Health will conclude the session. So we are doing it in a very sharp way, and the purpose is very clear. All of us must be enabled to answer any questions which is coming from our colleagues, media, and we can assure them that this is the situation and this is what we have to do. In fact, tomorrow I have to go for a long journey, and people are asking me, why don't you start wearing masks? Then I said, we have a discussion today, and I will decide whether I should wear a mask or not. So this is like, situation is like this. All of us are worried what we should do. And of course, we will wash the hands, but then mask, I will decide today. So with these few words, I invite Dr. Sham, because Sham, I understand he is in the office or he's on the way to the office, and he is cutting his work there, his clinical work. But because of his affection to us, he has come. Sham, kindly start.
Thank you so much. First of all, let me thank KUHS and the School of Public Health for arranging this webinar. So you have a great speaker lineup here. A lot of the people are going into the details of virology, epidemiology, public health, and diagnostics. My role here is to talk a little bit about some of the concerns, addressing some of the concerns from a big picture point of view of the metapneumovirus and the potential for pandemic. I know the concerns is that we're just coming out of pandemic and we are getting all this information from China. Those are the two major concerns that we have, because there are a lot of questions about the previous pandemic still not unanswered here. So I was going to talk a little bit about broad picture about what is the HMPV, what do we know about the current epidemiology and natural history briefly, and should we worry about the recent surges that we are being reported? And are there any good news about this virus? Because are we starting from scratch, or do we already have some weapons all over here? And what is the how does it lead to a pandemic? So what is the pandemic potential of that? So virologists in general are always hunting for new viruses. We always look at people who have symptoms similar to viral syndromes, and we collect samples from them and look for unidentified viruses. And so this is one of our friend and colleague, Ab Osterhaus, when he was in Netherlands in 2001. He collected samples from 28 kids who had a respiratory infection, not diagnosed by anything that known at that time. These samples were collected over about 20 years. And these people had, most of these kids have diseases from common cold, like early sniffles, all the way to people who were intubated for respiratory failure. So what we did, we took nasopharyngeal samples and then cultured them in different cell types. And in a tertiary monkey kidney cell line, it grew. And it grew, it had cytopathic effects similar to what you expect for a paramyxovirus. It's similar to the syncytium formation like we see for RSV, which is a prominent member of this group. And then he further characterized them and then looked at it, and the electron microscopy figures show here. This is the classic feature, if you're familiar with the electron microscopy of a paramyxovirus. It's very pleomorphic, and you can see this budding and like parts of the envelope coming out of this. By phylogenetic analysis, you can just see these clusters isolated. I want to talk to you a couple of things. One of the things is that it is very tight, right? These samples are collected over 20 years, and the genetic variability is not something that you expect for other similar things that we have seen recently, like coronavirus. Paramyxoviruses in general are not very highly variable. What that means that the people are going to ask, hey, do they mutate? Of course they mutate, but it's relatively speaking, they're actually not. The prominent members like mumps and measles, we have used the same vaccine for decades with great success. And even from most of you know from your experience with Nipah, which is also paramyxoviruses, the variation that you see from the 2018 all the way to the 3 or 4 pandemics are not great as you expect from a Delta to the Omicron type things coronavirus. So in general, they actually have limited variability compared to other particular viruses. So the structure of the paramyxoviruses is similar to most other components like RSV. It has a genome, it's an enveloped virus. It is a genome, is a single-stranded negative sense RNA. and it replicates primarily in the cytoplasm. It has 9 components of proteins here, and most of the proteins we talk about are the attachment glycoprotein G and the fusion protein F. These are the 2 proteins that are important in binding and targeting into the cells and getting into the host cells. This structure and the concept of the structure is very similar to what you expect with an RSV as well. It does not have hemagglutinin. That's a characteristic of paramyxoviruses, RSV, and metapneumoviruses compared to influenza, that it does not have a hemagglutinin function at all. There are some degree of variation appreciated globally. Genotypes A and B have been described, and clades 1 and 2 is subdivided. Those have been distributed across the globe over time. And fusion protein is something that I'll come back and talk a little bit about it. That's the protein of interest, similar to what the spike protein for COVID is there. So it's as an RNA virus, it gets into the cell and it uncoats itself and then moves into the cytoplasm. It uses the negative strand and RNA-dependent polymerase to make a positive strand, then translate into different components, and they get excreted. While doing that, it triggered two important aspects of the immune system. The one aspect is that it triggers a TLR7 type response that mediates interferon response and type 1 interferon. This is a protective response that all RNA viruses do, and you develop a response of interferon that will protect the cells from getting infected and limiting and recovery from the infection. But you can also do another pathway, TLR3 mechanism, to MYD88 pathway and NF-kappa B that leads to inflammatory cytokine secretion. This is also common with many RNA viruses. You know that with SARS-CoV-2 as well. And this is partly responsible for the pathogenesis of the disease that leads to ARDS and other functions, etc. But the amount of inflammatory response of metapneumovirus releases is much less than RSV and influenza in their group. So it is much less. However, when people look at it, if you take C57 black 6 mice and infect them with metapneumovirus, and you also do another group of mice and then mutate, delete the MYD88 response, what you find is that you don't really get the pulmonary pathology. So that means that this is a pathway that really leads into the pulmonary pathway. It's a host response to the metapneumovirus that leads to the pulmonary pathway. So this is the general theme why some people will develop disease pattern as well. The second part of virology I want to talk a little bit about is the attachment of the fusion. The fusion protein is important. It exists as a trimer, and it binds to receptor heparin sulfate. There are other components like beta-1 and integrin that's important in being the permissive for the host cell to replicate this particular virus. But this viral membrane just fuses with the host membrane to get entry into the cell. And by doing that, it binds to the cell, and then it changes its transformation, become this refolding and this really elongated form, and that purges into the cell, and this get into the cell for further replication to the cell. This is quite unique. This is similar for RSV, and there are some bacteria like also do that. Pertussis is another one example is do that. Most of these people have used that. And one of the things I wanted to tell you about this is about neutralizing antibodies. Neutralizing antibodies are important for paramyxoviruses because RSV, we know that we use neutralizing antibodies to prevent RSV infection. And that's been pembrolizumab is approved for using in patients who undergo hematopoietic stem cell transplant for the first 100 years, for 100 days. And these are all the sites of the protein, of the pre-fusion protein that's again. In the post-fusion, it changes its configuration, and you become more elongated. For RSV, the G protein and the F protein can generate antibodies that can be neutralizing. For HMPV, the G protein does not protect. Antibodies against G protein does not protect. What it protects is against F protein. And when it changes its configuration, these are the sites that actually generate monoclonal antibody responses that could lead to neutralization. And that's something to think about because neutralizing antibodies are being developed for therapeutics of this. So the crux of the matter is what do we know about what's happening right now? So when I get worried when people say that there is a new virus coming in, of course, there are concerns about what's happening in China. We have limited knowledge of what's happening in China also. So while our Osterhaus group discovered this in 2001, if those samples were from many years, and you actually have gone back and looked at samples from up to 60 years from now, and you can actually detect HMPV in those samples. So this is not a new virus. For example, HIV was discovered in 1983, but there are samples from like 60 years before actually had HIV in the samples from Cameroon and parts of Africa. So these exist. If you don't look for it, we don't really see it. So the news report saying that the first time we discovered finding this is there, and Dr. Srikumar will explain his experience about that. It is something that is already there. and it is the second most common respiratory pathogen in children. So if you take 100 kids in most global studies, the most common one in the winter times will be RSV, followed by HMPV, and then influenza. And that's those numbers can vary a little bit. In bad years, they can change, but that is generally the pattern of if you screen this data. And what is the mortality? This is the large study that is published about the natural history from globally of people who had complicated disease of HMPV. Not everyone with HMPV led to pneumonia or lower respiratory tract infection. 11 million people had reported in 2018, and of 500,000 people were hospitalized. Majority of these hospitalizations were infants less than 1 year, and that led to about 1,100 deaths. Most of the people who died, and this 64% of them are actually young infants and less than 6 months of age. And attributable, the paper concludes to show that many of these this is the global data, and most of these death comes from lower income and lower middle income countries, where most of the support measures of treating NICUs and putting ventilator support for young kids are not available. And this supportive care is one of the reasons for the mortality, responsible for the mortality, even though it is the mortality is like about less than about 1% mortality that we expect. So the next question is that, do we really have a surge? We have no numbers from anywhere, and we will probably not get any numbers exactly, other than reports saying that there is numbers are surging. Then if the countries who actually do surveillance of viral infections, such as the United States, the CDC does accumulate numbers, but they are not released in real time. Usually it takes a long time. And Dr. Srikumar can tell you that you have to isolate these samples and then analyze the samples and then say, what are these infections? So usually about like a year later or 6 months later. So it's not useful in real time. So we just cannot really understand the surge. And if you actually, most places are actually not screening for HMPV. In some countries, like in our country, in our hospital, we use what we call a biofire that will look for about 22 different respiratory pathogens. Someone comes to the casualty emergency room, that's what you get. Then you get all this data, so that is collected, and most of these are not treated, most of that not a major disease, but you can get data. But many people actually do not do that kind of wide variety of surveillance. It is not a new virus, and that's something that we should really tell people that this is not something new. And again, it's very common. It's already occurring, and mostly in kids and adults. An infection can give you partial protection. So the second infection you get will be much milder. And that explains why adults usually have mild disease, particularly the healthy adults, and may not be aware that they actually do have infection. We have to be concerned about adaptability. Paramyxoviruses in general are not very limited genetic variability. So they are not like orthomyxoviruses. They actually can change their genome and generate pandemic potential. They usually are not the one likely to do that. They are going to be there. They are going to have side effects in this subgroup of people, but they're actually the limited pandemic potential than other viruses. The mortality I talked to you about is limited to the infants, and the supportive care is important, and immunocompromised host. No specific antiviral that will be descriptive Latin. Ribavirin is the one that's been recommended as a pan-RNA virus antiviral. Somebody who's used ribavirin extensively is not a drug that most people favor. There are fusion protein neutralizing antibodies definitely available, particularly to the F protein that is really available. One of the things we learned from COVID-19 is that you can generate neutralizing antibodies and bring them to market very quickly. Companies like Regeneron can do that in like 4 to 6 weeks. So that's definitely a potential there. There is also a vaccine, a combination mRNA vaccine from Moderna that has undergone phase 1 study, moving on to phase 2 study. It is a combination mRNA sequence that covers parainfluenza 3 as well as HMPV. So that's something, it's already there. The safety is proven. There are other candidates in live strains also being developed. There is no huge development in this area because it's a milder disease and particularly targeting small number of patient population. It is the needs is not the highest in terms of vaccination. But you know that RSV, generally last year we had a vaccine that's approved. So there is a trend that many of these respiratory viruses, the vaccination will be available pretty soon. So the last question is that, if you have to put a bet, paramyxoviruses are not the most common ones to have great variability. and infect a lot of people. The R naught for RSV is about 3, R naught for measles is 25. You know, the Omicron it was like 8 or 9 in its peak, and the R naught values for HMPV is much lower. However, we need to trend. The changes in behavior of these viruses need to be monitored. Any particular thing that had happened. However, it's unlikely for a Metapneumovirus, knowing what we know, to be able to gain that kind of variability moving forward. Generally, viruses are more concerned about pandemic potential because they replicate much higher than any other microorganism. So, and RNA viruses are more problematic than DNA viruses because they use cytoplasmic capacity to replicate, and they actually can replicate much faster. Their host restrictive elements are less effective against RNA than DNA viruses. Segmental genome organization is something we worry about. Orthomyxoviruses, like we talk about antigenic shift, etc., with influenzas, is something that we always concern. Big chunks of DNA can be replaced, and then all of a sudden you have a new influenza virus. That is always a threat. And we all, as providers, we always be concerned about those much more than HMPV related thing. Small genome sizes are more adaptable to mutations, and the bigger the genomes, like the pox viruses, don't really have a lot of mutation capacity. They do mutate, but you don't, and paramyxoviruses are in the same medium size, they usually don't mutate as well. Respiratory route of transmission is something that's important. That's for a pandemic potential, that's a key element because you can spread much faster. And that's the key element of HMPV transmission. That's something we're concerned. But other than that, you know, if we had to put money on what we know already, the HMPV related pandemic potential is actually quite low. Virologists, you know, like me, would be more concerned of avian flu because we already talk about, you know, remains the major threat, or a seasonal influenza with an antigenic shift will become a major thing. So our job is to reassure people this is not a new virus. It's usually present. It's very common in kids. And in adults where it causes asymptomatic infection. And there is nothing that we know so far that there is a change in the behavior pattern that will lead to a higher mortality, higher transmission, or pandemic potential. Thank you.
Thank you, Professor Shyam, for the excellent presentation. Now, Dr. Shyam has talked about the virus, the discovery of the virus, and saying that it is there even in the samples from 60 years ago. And so proving that, and our first question that whether this is a new virus or not. So it is not a new virus. Then he has talked about the pandemic potential also. The pandemic potential is low, but we have to be careful and we have to be monitoring also. That is the lesson we have learned, also the lesson we have learned. Then you have also talked about the difference between and the similarities between the RSV virus and also the HMPV virus. Now I think we can go ahead with the questions. I think there is two questions there in the chat box. Dr. Purushothaman has asked a question. Is the fusion protein very much related to RSV, so that mortality in young infants and immunocompromised hosts can benefit from monoclonal antibodies, recent ones, and the vaccines, which is recommended in pregnant ladies in developed countries?
So, yeah, so structurally fusion proteins are similar, but antigenically they're different. So you cannot really use a monoclonal antibody raised against RSV like pembrolizumab to treat patients with HMPV. I believe that's what Purushothaman, sorry, your question was. So you do have to develop monoclonal antibodies against this particular agent and be able to show that that's actually neutralizing capacities there. That's having said that, that's really not a tough task. For all the things we could do, and I believe that's one of the best things that happened in COVID was development of monoclonal antibodies that saved a lot of people getting infected. That is something, a low-hanging fruit, if this becomes an issue. But I'm not really concerned that this is a major concern with a lot of people coming in, rather than we already see in our hospital system.
Purushothaman sir, you could be proud that Dr. Shyam Kutlil is an alumnus of Trichur Medical College. So next question is whether syrup oseltamivir helps in treating HMPV infection.
Right, another good question. So oseltamivir or Tamiflu was developed for an activity against influenza. It has, whether it has activity in other things, I personally believe that it has very limited activity in influenza itself, because the studies were done in a way that makes you feel better a little bit earlier. It really have no clear clinical endpoints, no change in mortality, etc. We use them extensively without a lot of randomized control studies in immunocompromised people and host. So generally it has activity. Whether it has activity in HMPV in a way that it will have any public health importance is not known at this time.
Any other questions? There are only 2 questions in the chat box. If there is any other pressing question. I think there are no more questions. I would like to thank for the Kerala University of Health Sciences and also all the people who are present here for giving this excellent session where we have learned a lot. Thank you for being here at this hour of the day and with all the difficulties with the weather and the work and all that. Thank you, Dr. Professor
No
Sam
problem.
Kotelel.
I just last thing I say, I put my email there. If you have any questions, please email me. Thank you so much. Nice to
Okay.
see you all.
Thank you.
Thank you.
Suma, one second, Suma.
Yes, please.
I take this opportunity to welcome Murali sir. Murali sir is our senior most teacher. Sir has come, sir. Most welcome, sir. Thank you. We all seek your blessings. After a long time, I saw you. Thank you very much.
No, one more thing is that you only raised a little while ago regarding wearing of mask. Mohan,
Yeah, yes, sir.
what is the
Can you reply, Shyam?
using a mask as a preventive measure at this point of time to prevent the spread?
So a lot of people, thanks, Murali sir. Very nice to see you after a long time. So there is, the mask, whether the mask is useful right now, that's actually not indicated. There is no reason to say that because the infection rate is not. But people do wear mask. If you're an immunocompromised person, of course, a lot of people have been wearing masks much more than before the pre-pandemic era. But whether the mask will definitely, I'm one of the people who believe that mask will offer some degree of protection, particularly the person who may be symptomatic. And that's the way many of these masks are constructed. So that will allow you to get the aerosol distribution a lot better. So not so much for the recipient, and that's definitely there. However, there is no guidelines to move forward in terms of using mask at this time, because the number of cases is still not alarmingly high. And individually, you will gonna have to develop data on our own, seeing that how it is going up or not. A lot of the panic is in the social media and the news media because they're looking for sensational news, and this is great because it's coming from China soon after the pandemic. I don't think it is that concerning at this time.
Thank you.
Thanks.
Yeah,
Thank you.
Thank you, Shyam, for coming at a very short notice. Thank you so much.
Thank you, Murali sir. I think we can move on to the next session. The next session is on clinical features and management, including prevention of HMPV infection by Dr. Arvind Kumar. Arvind is, I know all of you know Dr. Arvind. He's the head of the Department of Infectious Disease at Medical College Trivandrum. And he's always a sort of type of person since 2019, whenever there is an infection, he used to help everyone, including the government, in development of guidelines and now even policies. So over to you, Dr. Arvind, for the clinical features and management of HMPV infections.
Thank you, Madam, for the kind introduction. At the outset, I would like to thank Honorable Vice Chancellor Mohan Sir, Rajmohan Sir, and their team from Public Health, School of Public Health for giving me this opportunity. So next 15 minutes, I'll be taking you through the clinical presentation. Madam, the slides are visible, right, Madam?
Yeah, right, sir.
Please put it in the slide mode. Is it
Okay.
present there?
So next 10 to 15 minutes, I'll be discussing about the clinical features and the management options of human metapneumovirus infection. So basically, Shyam sir did set the background for this presentation. We all know that human metapneumovirus is not a new virus, and it usually leads to upper and lower respiratory tract infections of all age groups, especially symptomatic diseases seen in younger children, those less than 1 year especially, and in older adults, above 65 years, and those who are immunocompromised. In children with acute respiratory tract infection, human metapneumovirus is, according to many studies, is detected in up to 5 to 15%, and usually seen most commonly in children less than 1 year. The seroprevalence is almost 90%. So that means that when an adult gets infected, it's more often a reinfection. And Sharda madam will take us through the different genotypes, and the immunity that we gain is actually transient. So there is always a chance of reinfection, but the severity is likely to be very much less. So I have actually selected studies dating back to 2004 just to show that this is not a new virus. So a lot of literature is already available on the symptomatology of human metapneumovirus, especially in healthy infants and children. So this is from an NEGM study in 2004. And this was a large prospective study in children with lower respiratory tract infections, where the human metapneumovirus was identified in 20% of the samples, which were negative to other pathogenic viruses. And the mean age of the infected children was 11.6 months. And the major symptom noted was actually bronchiolitis. And 59% had symptoms of bronchiolitis. The virus was isolated only rarely from asymptomatic children. Human metapneumovirus infection is a leading cause of respiratory tract infection in young children. So this is an observation which was made way back in 2004. And the conclusion is that the symptomatology is almost similar to that of respiratory syncytial virus infection, which is a very, very similar virus to human metapneumovirus infection. And the seasonal epidemiology is usually it is peaks during the winter season and leading up to the spring season. So basically, all these viruses lead to acute respiratory tract infections in children, metapneumovirus, RSV, influenza, parainfluenza, and adenovirus. And usually the metapneumovirus is detected during the winter season up to the spring season, a season where you can see a lot of surge in respiratory syncytial virus as well. And we know that in China also the major the pathogen is influenza. 30% of the isolates are supposed to be influenza. It's only because human metapneumovirus appears to be something which is unfamiliar to the media. So this UN cry is centered around human metapneumovirus. And looking at the symptomatology in children from the same article, cough is seen around 90%, coryza in 88%. Just like in RSV, the fever is there only in around 50% of the affected children. Then wheezing is there in, as a symptom in 22% and as a sign in around 52% of the children. And when we look at the symptomatology from 3 case series, it's seen that the cough is the predominant symptom in children as well as in adults. Around 68 to 90% will have the cough. Rhinitis in around 44 to 77%. Fever in around say 50 to 60%, and wheezing in around 51 to 56%. So basically, it means that just like an RSV or other respiratory viruses, an acute respiratory infection which can lead to bronchiolitis or wheezing, one of the DDs we need to think about is human metapneumovirus. In the same study, these slides take us through the clinical diagnosis of lower respiratory tract infections caused by human metapneumovirus as compared with other respiratory viruses. The compared viruses are RSV, parainfluenza, influenza, and adenovirus. And we can see that around 59% of the admitted children had bronchiolitis compared to 65% in RSV. The group was present in 18%, whereas in parainfluenza, the group was there in 64%. And the most important aspect is the percentage of the patients, admitted percentage who had pneumonia. So of the viruses studied, the human metapneumovirus was the virus associated with the least percentage of development of pneumonia. It is 8% compared to 28% in influenza, 21% in RSV. In parainfluenza also, the percentage of pneumonia is very less. But the thing is that, when you look at the pathophysiology, why, the metapneumovirus leads to symptomatology, as Shyam sir did allude to, the receptor is the epithelial receptor. There is an integrin. This is alpha beta integrin. There is a receptor. And this virus lead to the hyperproduction of mucus, as well as in hyperresponsiveness to any stimuli, along with an epithelial hyperplasia. And that is the reason why the children are likely to develop the wheezing episodes as well as exacerbation of asthma, which is observed in adults with COPD as well as asthma. And this is a study from Taiwan. This is also from 2006. So, which actually compared the clinical manifestation of metapneumovirus with other respiratory viruses leading to acute respiratory tract infection and influenza-like illness. This actually shows that, the previous study shows that clinically, based on the clinical features alone, it's not easy to differentiate between human metapneumovirus and other respiratory viruses. Even the presence of rash is there in metapneumovirus in around 5 to 10% of cases, but that can be seen in other respiratory viruses as well. So based on the clinical features alone, you cannot differentiate human metapneumovirus from other causes of acute respiratory infection. When we look at the lab parameters also, there is not much difference between the lab parameters observed in a metapneumovirus infection from that of other respiratory virus infection. So the WBC count, etc., based on this alone, you cannot differentiate between a viral or a bacterial thing. The interesting thing is there is a lymphocytopenia which is observed in the early part in almost all the virus infection. In other respiratory virus infection, the lymphocyte count decline was slightly higher than that observed in metapneumovirus infection. So all the other parameters were actually relatively same in other respiratory virus infections compared to that observed in metapneumovirus infection. When we are looking at the radiological aspects also, there is not much difference between other respiratory virus infections and the metapneumovirus infection. So what we actually see is bronchiolitis, that is a perihilar infiltrates, hyperinflation, then to an extent atelectasis, and some children can develop the consolidation as well. The presence of pleural effusion and pneumothorax are features which point against a diagnosis of metapneumovirus infection. And you can see that just like what was depicted in NEGM from US. So there is always a co-circulation of all the respiratory viruses like metapneumovirus, influenza, RSV, adeno, influenza B, etc. So without a molecular diagnostic testing, it is very difficult to differentiate between these pathogens as a causative agent for acute respiratory infection. So this slide was actually used to as part of the state guidelines which were released last year. That is, we had a co-circulation of COVID as well as influenza, and we had also RSV as well. So this slide we used to communicate to the doctors at the ground level that any patient who is presenting with influenza-like illness, we need to think about COVID-19, influenza, RSV as well as adenovirus. So and that is how the term triple demic which came about, that is COVID, influenza and RSV. Influenza, we all know it could be B, influenza A, H1N1, H3, N2, etc. And in the current context, if someone is presenting with the same symptoms, you need to understand that it could be human metapneumovirus as well. And we are more worried about this mycoplasma. So emergence of, macrolide resistant mycoplasma, and actually the state guidelines for the macrolide resistant mycoplasma will be released in a couple of weeks. And this metapneumovirus actually came into the media just because it is not a virus which is known to the majority of the media people like that. So I think that, as Srikumar sir will take us through the viruses and bacteria covered in the multiplex PCR panels, and all the doctors should be familiar with all the names there. So human metapneumovirus is there, Boca virus is there, all the human coronaviruses are there. So something is there in a multiplex PCR panel means that it is not a new virus. It's a very common virus, which is a common etiology of a respiratory tract infection. That is something that all of us need to keep in mind. So in the current context, if someone is actually, whether it be a child or an adult who is presenting with acute respiratory tract infection or ILI, what we need to follow is the existing state guideline of a combined ABC. So the aim is to identify whether the patient requires treatment or not. So the basic concept of this combined ABC guidelines is that based on the severity of the symptoms, we need to categorize into A, B1, B2. B2 is those with comorbidities, and C. The aim is to identify patients with the category C symptoms, that is those with complications like pneumonia, dyspnea, and all those things. They require admission. And this category which requires molecular testing, and based on the diagnosis need treatment. Metapneumovirus, we don't have any effective antivirals as of now, and majority of the infections don't require any specific treatment, but the supportive treatment is of paramount importance. Now, with regard to any respiratory tract infection, and for that matter, any infection, the most important thing a doctor should look at is a red flag sign. So even at the time of presentation, if the patient is not having that much of apparent red flag signs, we should educate the patient about the chance of development of red flag signs, so that they can come back to us when they if they develop those, like the presence of altered sensorium. Rarely, even in human metapneumovirus infection, the encephalitis is described in infants and all, very rarely. The presence of dyspnea, drowsiness, chest pain, hemoptysis, excessive fatigability, syncope, palpitation. So this is applicable to almost all the respiratory tract infection. And the complications that we are actually looking out for in human metapneumovirus infection, just like in RSV and all, is the bronchiolitis, exacerbation of pre-existing asthma, the development of pneumonia, as well as certain immunocompromised patients or very young infants and all can go in for ARDS as well. Now, this is a checklist that we actually piloted during the COVID-19 time. That is, now if you are sending back patients with category A symptoms, the thing is that they are actually kept under follow-up. So based on this checklist, they can know whether when they need to come back to the hospital, and this applies to almost all the respiratory tract infections. So basically, what we expect in the primary care center, etc., is that any patient with ARI, at the time of presentation, if they don't require any this thing specific treatment per se, they should be educated about the red flag signs and when they need to come back to the hospitals for further evaluation. And if they develop any category C symptoms and all, definitely admission as well as molecular diagnosis has to be performed. Now, coming to the transmission dynamics and the prevention aspect, the incubation period is around 5 to 9 days. And what we need to adopt is the contact and droplet precautions, which is common to all the respiratory viral infection. The good thing is that it's an enveloped virus. Enveloped virus means that the soap and water is very effective. So just by hand hygiene, so the virus in our hands, etc., can be destroyed by the soap and water. That can be done. And this metapneumovirus can lead to outbreaks in healthcare facilities, and studies have shown that in transplant centers, the neonatal oncology center, long-term care facilities, etc., the outbreaks have occurred. And the secondary attack rate is supposed to be around 34 to 72%. And the vaccines, as of now, we don't have any vaccines, but we have a lot of vaccine candidates in the pipeline based on the F protein. Now, when we come to the adults, the people above 65 years of age and people who are heavily immunocompromised, like lung transplant recipients, etc., metapneumovirus is actually a problem. So this study is very, very important. This actually was done in hematopoietic stem cell transplant recipients and looked at the factors associated with progression to lower respiratory tract disease. So this study showed that around 5% of the patients have upper respiratory tract infection due to human metapneumovirus infection following lung transplant, following the hematopoietic stem cell transplant. And then it looked at the factors which led to the progression to lower respiratory tract infection. That 60% of them did progress to a lower respiratory tract infection with a mortality of around 20%. So the factors which are associated with progression to the lower respiratory tract infection or pneumonia in this study were actually the host-related factors, like the lymphocytopenia. The lower the lymphocyte count, the more the risk of progression to lower respiratory tract infection, as well as the cumulative dose of steroids used. The patients were on more than 1 mg per kilogram of prednisolone, the chance of progression to pneumonia was actually higher. This is very, very important because one factor which was clearly studied was that the viral load in the specimen did not actually correlate with the development of lower respiratory tract infection. So basically that means that the pan-RNA viral drugs like say ribavirin and remdesivir may not be really needed. What is required to treat even in heavily immunocompromised patients is we need to modulate the host factors, like we may have to bring down the immunosuppression, the use the steroid dose like that, like what we do in BK virus infection. So another problem is the human metapneumovirus infection, lung transplant recipients. In lung transplant recipients, in those with metapneumovirus infection, the chance of a chronic lung allograft dysfunction occurring at 1 year is around 33%. So this slide clearly shows that the human metapneumovirus infection leading to complications is actually related to the host factors rather than the viral factors. Even in a heavily immunocompromised patient, the way forward might be modulating the immunosuppression. And still, majority of these studies were done in the pre-COVID era. And this study from Spain shows that we should be actually on the lookout for whether there is any change in the clinical presentation, severity, etc. This study was done during the 6th wave of COVID in Spain during 2021. So what they observed is that they compared around 48 patients with the children with metapneumovirus infection in 2021 with around 500 children. This is a retrospective cohort from 2005 to 2020. What they found is that in that outbreak, the age was slightly higher than usual, and the patients developed more hypoxia and pneumonia, longer length of stay, and greater need of intensive care. We need to understand that this study was done during the COVID era. Okay. And we can see that the duration of stay in this study is slightly higher. Duration of fever is slightly higher compared to the historical cohort. Duration of hypoxia is more. Also, around 35% of the patients developed pneumonia in 2021 compared to around 8.8% who developed pneumonia between 2005 and 2020. So this is something that we actually need to be on the lookout. Another interesting aspect is that there is always a chance of a viral co-infection. So in this study, around 39.6% had co-infection with other viruses like RSV, COVID and all. And historical data also is around 40% is having the co-infection. So this is the reason why, we need to be, on the lookout for any change in the clinical presentation. As of now, WHO as well as, the global authorities have not said that there is any change in the clinical presentation of metapneumovirus, which is observed in China now. And there is, as Sham sir did say, we are not expecting a huge change in the genetic framework of human metapneumovirus as well. So basically, all this hue and cry is basically because human metapneumovirus appears to be a new virus, at least to the media and a subset of doctors, who are not really aware of that the human metapneumovirus has been existing for a lot of years. And that is the reason why, I would suggest that, when Sri Kumar sir takes us through the BioFair panel, we should be familiar with each and every virus listed out there. That means that, Boca virus infection is here for quite some time. But suppose, in China starts surveillance for Boca virus, then media will pick it up and it might become a huge story. Regarding the preventive aspect, whether, we should be wearing mask and all, the state policy right from 2022 December is that any patient who is at risk of development of a severe disease due to any virus, like those patients with uncontrolled diabetes, immunocompromised, transplant recipients, those on dialysis, etc. Ideally, they should be wearing mask when they are going for 3C settings. 3C means, closed situations like when they are traveling in a lift or something like that. Then close contact settings when they are coming into contact with another person within 6 feet, and a crowded place when they are going into a marketplace like that. In these situations, it is always ideal for a patient who is likely to develop a severe disease due to any respiratory viral infection to wear mask, and obviously hand hygiene goes hand in hand with that. So thank you very much for the opportunity.
Thank you, Dr. Arvind, for the elaborate discussion on and a very clear discussion on the clinical features and complications and the comparisons or the difference from the RSV virus and also the influenza virus, so that in children, it may be difficult even to clinically diagnose HMPV. And also, you have specifically pointed out that it's the highest risk factors are at the host, that is especially the transplant population, transplant of different organs, because nowadays the people who are undergoing transplantation is increasing. And so we have to be very careful with children as well as elderly and also the immunocompromised people. It was a very good session and a lot of learning it has been transferred. Thank you, Arvind. I think we'll have the discussions after we have finished all the 3 sessions, because there could be a common the questions where all the 3 could be answering. Thank you, Dr. Arvind. And now we'll move on to the next expert. Next expert is Dr. Sharda. Dr. Sharda is the professor and head of Department of Microbiology at SUT Hospital now. And previously, she was the head of the Department of Microbiology Department at Trivandrum Medical College. And Dr. Sharda has worked a lot and in developing the testing and other things during the COVID, and as well as in the related to the antimicrobial resistance. So we'll hear from Dr. Sharda on the virus and its details about the virus. Over to you, Dr. Sharda.
The slides are visible, madam?
Human metapneumovirus. Thank you.
Am I audible?
Yes, very well.
Okay. Thank you so much for the invitation for this presentation. Honorable VC, Rajmohan sir, and all others of the faculty members. It has made, Dr. Sham Kotilil has made easy for me because I need not talk anything about the replication of the virus. So just now we'll go to the virus. That is, as everybody has told, Aravind, Dr. Sham has told, it was described in 2001, but at that time itself, there was serological evidence of extensive pediatric infection dating back more than 50 years. And so human metapneumovirus is newly discovered rather than newly emerged. And we all know that human metapneumovirus takes a long time to grow in vitro. And it has got a delayed cytopathic effect, and it requires added trypsin for the activation of the fusion protein. So it's for growing it and studying it, it's very difficult because it takes a long time. And so now we have to remember is that human metapneumovirus is newly discovered rather than newly emerged. It was there. Human metapneumovirus was there. So it is an important agent of respiratory tract disease, particularly in now Dr. Aravind has already told us that in pediatric and elderly population, but its impact is less than that of RSV. So we'll see about the virus as such. It belongs to the order Mononagavirales and family. Now it has been International Committee on Taxonomy of Viruses have now separated into a separate family. There is a pneumoviridae, and we have two genera, orthopneumovirus and metapneumovirus are there. Orthopneumovirus, you have RSV and a number of animal pathogens. While metapneumovirus, you have your human metapneumovirus and avian pneumovirus. So this is the classification of your human metapneumovirus. So this, Dr. Sham has already told, it is a highly pleomorphic virus, and it is a single-stranded RNA virus of negative sense. When you look under the negative stain electron micrograph of human metapneumovirus, you have seen that it has got spikes on its surface, and sometimes it may be seen not only as spherical, filamentous also. And about the genome, just a few words about the genome. He has already told that it has about 8 genes and coding for 9 proteins. So you have your N is the nucleoprotein, P is the phosphoprotein, M is the matrix protein, F fusion protein, and M2 you have your transcription elongation factor M21 and RNA synthesis regulating factor M22. Then you have your SH. SH is the small hydrophobic protein. G is the glycoprotein, and L is the polymerase protein. So that is the genome of, it's about 13 kilobases, and this contains about 8 genes coding for 9 proteins, because you have M2, you have M21 and M22. M21 is the transcription elongation factor, and M22 is the RNA synthesis regulating factor. So what is the difference between this RSV and human metapneumovirus genome? There is some difference. You can see here, NS1 and NS2 is the non-structural proteins that you see in RSV, but it is absent in human metapneumovirus. Because this non-structural proteins, NS1 and NS2, are interferon inhibiting genes. Interferon inhibiting genes, these NS1 and NS2, which the human metapneumovirus does not have. And one more difference is that in the order of the genes, you can see the genome. Can you see N, P, M, F, M2, SH, G, and L, while here the order is different of the genes, and absence of NS1 and NS2. And very important is because the non-structural interferon inhibiting genes are absent in human metapneumovirus, they are interferon inhibiting NS1 and NS2. Why? That is why RSV is more virulent, or it produces more serious effects than human metapneumovirus. While human metapneumovirus does not have that NS1 and NS2 non-structural proteins. I'm not going to the replication because Dr. Sham has told about it, because it's a negative stranded. I've just taken it off replication because it is a single stranded RNA virus of negative sense. You can see it has got spikes on its surface, and the fusion protein attaches to the integrin, and it enters by clathrin mediated endocytosis. Then because it's a negative strand virus, it has got an RNA-dependent RNA polymerase. Transcription translation occurs, and the virus buds out, and thus it's an enveloped virus. Now it has got two major genotypes, A and B. Human virus has got two major genotypes, A and B. It has 4 subgroups, that is A1, A2, B1 and B2, several sublineages. A2 has got A2, AB, and now in China, they have got A2C, they have got it. So there is not much difference in all these sublineages in regard to the seriousness of the disease or severity of the disease. There's not much difference. So you have major genotypes is A and B, and when it is sequenced, we can see that, we have seen that there is amino acid identities between the genotypes where 80 and 90% respectively. So this is an RNA virus. So it will undergo some, it's not a DNA virus, since it's an RNA virus, there will be some mutation occurring, but it will never go into a pandemic as or COVID-19. It's not a new virus, and everywhere in India, in Kerala, we are doing testing for, Dr. Srikumar will be talking about it, not only in Trivandrum itself, many places, but we have the prestigious Institute of Advanced Virology where they do all the viruses. We have got in RCC, in Sri Chitra, or because BioFire is very expensive. As Dr. Sham has told, we have got that panel, respiratory panel. It is very, very expensive. That is why we are not doing it as frequently as as in the developed countries. Again, this is a difference between the orthomyxoviruses, that it is a segmented virus. Here it is not a segmented. You can see it's a non-segmented. It's a helical nucleic acid, and the envelope, you can see we are seeing spikes on the surface. They are the envelope glycoproteins spikes. The fusion protein is very important because it is attaching to the receptor, and that is how the virus is entering by clathrin-mediated endocytosis. And you can see in the nucleocapsid, you have got the nucleoprotein, phosphoprotein, and you have the M21. I've told you, you have got transcription elongation factor M21 and RNA synthesis regulatory factor M22. And these are the, in short, about the virus. And now we'll see about the spikes. These are the 3 very important. F is the fusion protein. It is the neutralization and protective antigen. And you have got the, in the genome, you have got nucleoprotein, phosphoprotein, and polymerase. And you can see M21 and M22. So M22 is very important. It is a RNA synthesis regulatory factor, while M21 is a transcription elongation factor. Okay. And as these viruses can be, for the first one, I have shown is RSV, which produces syncytium, while human metapneumovirus does not produce syncytium as RSV, but you can see rounding of the cells. It's a monkey kidney cell, LLC MK2 cells, monkey kidney cell line, it will grow. And you can also take the centrifugation culture and stain with monoclonal antibody 8, you can see, and do an immunofluorescence also, you can see. The, in fact, human metapneumovirus infected culture can be demonstrated. So it takes some time to grow in these cell lines, that is monkey kidney cell lines. So you have to do either a shell wall culture can be done and fast you can grow it. Otherwise, this infected LLC MK2 culture can be taken and stained with the monoclonal antibody 8. Transmission. As it is spread through contact with contaminated respiratory secretions, droplets from coughing or sneezing, and the virus can also survive on the droplets for about 6 hours. It can survive, and it can contaminate the surroundings while coughing, sneezing, and for allowing for indirect transmission. So it's very similar how to prevent. Prevention also, Dr. Aravind has told. I'm not going to the prevention side because, as he told, that you have to wear mask if you are an elderly, over 65 years, and you are coming into contact with a patient with respiratory infection. It's better that you wear mask and washing hands because it's an enveloped virus. Wash hands with soap and water, and the contaminated can contaminate for about, like COVID, where it can survive in the droplets for about 6 hours, and it will survive and it can transmit also. So the surfaces also has to be cleaned where the patient is there. So wearing mask for the patient as well as for the doctor is very important when you see these patients, because it can transmit the infection. And one more thing is that I have to tell is that this virus is not a new virus. It is here in India, in Kerala. And now Dr. Srikumar is going to tell about the statistics. Why I'm telling, it's not a newly discovered virus in 2001. It is here we have this virus here. So prevention is very similar what Dr. Arvind has already told.
ഇവിടുത്തെ ഡയറക്ടറാണ്.
Okay, thank you. Thank you so much.
Thank you, Dr. Sharda. Excuse me, Dr. Anthony Kollanur, could you please mute? Thank you, Dr. Sharda, for the excellent presentation, where you have reiterated what has already been said by Dr. Shyam Kutilil, and also given the important differences between the RSV and the HMPV, and also that how to whether the importance of using the mask and other preventive measures. Thank you very much. Now we'll move on to the next session, the last session, that is on the Kerala experience with diagnostics by Dr. Sri Kumar, who is the director of the Advanced Virology Institute, Thonikal. And over to you.
Good evening to all. Am I audible?
Yes, you are audible.
And the
Very
slides
well.
are visible?
Yes.
Okay, so pardon me if I repeat many of the things that was said by the previous speakers, being the last presenter of the day.
No
so
problem.
maybe like I would like to recapitulate many things that was already said. So this is the first publication on human metapneumovirus, which was mentioned by Dr. Shyam Kotal in the beginning. And so this is the screening that they have done on 28 young children. And in fact, when they started looking at the etiological perspective of this particular disease, they had to do a lot of experiments to see that there is a new virus that is present. Essentially, it started with looking at the CP, which was a delayed CP, which they could get in a tertiary monkey kidney cells, then in Vero cells and A549 cells. Then they could also see that this virus that is present in these samples are able to infect the cynomegalovirus macaques and cause a disease. So thereby establishing the Koch's postulates. Then they had to do a random arbitrarily primed PCR based detection, a molecular detection method, which was using degenerate PCR primers to see that there is a virus present. A partial sequencing showed that it is a new virus, which has some relatedness to the RSV. And full genome sequencing could identify that it's a completely new virus, much related to RSV, but significantly different from RSV. So this was the initial studies. And later, there are a couple of studies looking at the cytopathic effect of this virus that is happening in different cell lines. I have shown an example on Vero cells and LLMCK2 cell lines, wherein they could see the delayed cytopathic effect. And as Dr. Sharda Madam was telling, it is not typical with the RSV style, but it's different, rounding of the cells and also the sloughing of the cell monolayer. But in different cells, they behave differently. And also sometimes it is difficult to isolate this virus much easily. Now, this is a simple classification scheme which was mentioned earlier. The Paramyxoviridae has two subfamilies, Paramyxovirinae and Pneumovirinae. And in the Paramyxovirinae group, we have all these major pathogens like Nipah, measles, and other related viruses. And the Pneumovirinae has the pneumovirus, that is the RSV, and the metapneumovirus, that is the human metapneumovirus and the avian metapneumovirus. This is the genomic structure, which I will not explain again. And these are the, as it was mentioned, the glycoprotein G and F are the major proteins of our interest many times, because these are the proteins which are used in the diagnostic purposes as well as also making the monoclonal antibodies. Now, one of the major discussions have been what is the origin of this human metapneumovirus. So if you look at the amino acid sequences and do a comparative analysis, you will see that it has probably an avian origin, and especially a very close relation with the turkey rhinotracheitis virus. So as we know, like all these pandemics that are emerging had an animal source, and even this metapneumovirus at some point of time had a previous source from a bird. So but fortunately, and also like we see that this metapneumovirus is also slowly evolving into sublineages. And now if you look at the world over, the variant A2B is the one that is spreading much faster. So there is some genomic advantage or the selection advantage also happening to these metapneumoviruses, but it may be slow than many other viruses. That is why we don't see a major surge in the infections. But definitely there are selections happening in the evolving viruses. But one of the major fortunate thing for us is there are no animal reservoirs as such. So the spillover infection, frequent spillover infection chances are limited, but there is some selection happening in these viruses. Now we have animal models to study this. That means that they can infect animals like small animal models like BALPC mice or C57BL6 mice. And non-human primates are also susceptible for studying the pathogenesis of this metapneumovirus. Now, one more important point is about the G protein. The human metapneumovirus G protein is the receptor binding protein, and it is the protein that shows the maximum variability. So between the HMPV subgroups. And when you look at the mutation rate of this particular protein, it is very interesting. The G protein or the gene coding for the G protein that has got a mutation rate of 3.5 into 10 raised to minus 3 nucleotide substitutions per site per year. That is the unit that they mention on the mutation rate. Compared to the F gene, which is 10 times less prone to mutation, that is 10 raised to minus 4. N gene is also 10 times less prone to the mutation. That means among the major genes like F and G, G is much prone to the mutation, and that is the receptor binding protein, and also that is the one that decides the transmissibility of the virus. And we should also know that the SARS-CoV-2, when it spilled over, it causes the initial pandemic in the beginning. The spike protein, which is the receptor binding protein of the SARS-CoV-2, had a similar mutation rate, that is 2.19 into 10 raised to minus 3. So comparing human metapneumovirus as well as the SARS-CoV-2, the major receptor binding protein shows similar kind of mutation rate. These are recently, some of the recent publications have compared the mutation rate of SARS-CoV-2 in the beginning of the pandemic as well as the late stages of the pandemic. And they could see that as the pandemic progresses, the mutation rate comes down. So this particular, I leave the possibility of a pandemic, the potential for causing a pandemic by the HMPV to the audience. But there is always, being an RNA virus, it always presents a potential for mutating into different variants. And as it was mentioned by some of the earlier speakers, our worry should be more on the clinical presentation, how this disease severity is changing, and whether this disease profile pattern is differing from the previous cases. Now, the global scenario, it's a global pathogen. It has been identified in every continent, and also many times co-infections happens. And also there is a seasonality in the infection. And as I mentioned earlier, the sublineage A2B is the major sublineage that is circulating now. And around 5 to 10% prevalence of this HMPV is there in worldwide. And in India, HMPV has been identified in India as a respiratory pathogen in 2004. I have quoted the publication from National Institute of Virology. So when it was announced that the first case of human metapneumovirus was identified in India, 2 days back in Bangalore, that was really surprising because already there are large number of publications on the human metapneumovirus from India itself. And also, we also have some idea about the average prevalence rate of this infection, like some 4 to 12%. And the circulating lineages have also been identified in India, like B1, B2, A2B, and A2C. So there have been significant studies or information on the metapneumovirus circulating in India. So as such, it is not a novel infection or novel virus that has been introduced. Now, the laboratory diagnosis, as I mentioned earlier, the G protein is the one that we focus on laboratory diagnosis, and serological diagnosis are not available. So the molecular diagnostic approaches are the one that is being used. The conventional reverse transcription PCR, that is RT-PCR, is possible. It takes around 5 hours, 3 to 5 hours to get the results. The sensitivity is comparatively low, like 1,000 copies per reaction. But when we move to the real-time quantitative PCR, the sensitivity is better. We can do it much faster. That is the assay that is the more widely done. But there are other assays like loop-mediated isothermal amplification, recombinase aided amplification, recombinase polymerase amplification, then metagenomic approaches, virus isolation. All these can be done, but each of these diagnostic approaches have got its own advantages and disadvantages. But the most easily available one or fast available one is the real-time quantitative RT-PCR. Which is being employed for most of the viral infections nowadays. Now, in our Institute of Advanced Virology, we have been testing HMPV since 2022, along with the other respiratory viruses from the nasopharyngeal and throat swabs using real-time RT-PCR. So the G gene is the target. And this is a typical panel that we do. As Aravind was telling, maybe we can note down the viruses that we are doing. We check around 22 pathogens, including mycoplasma and some of the bacteria like Staphylococcus and Streptococcus. And all the other viruses, including the metapneumovirus, Boca virus, paracovirus, and various coronaviruses, parainfluenza viruses. All these we test in our panel, and it is not as costly as the BioFire that we do in many institutions. So this is much affordable to the patients, and we get a lot of samples for getting tested. And during our testing in the last 3 years, we could pick up a number of human metapneumovirus cases. In 2022, we were beginning our diagnostic facility. We were getting only very limited number of samples. 191 respiratory samples were tested at that time. We could pick up 3 positive cases. 23, we could get 380 samples, and 1 positive case we could get. And 24, we tested around 4,000 samples and 8 positive cases. And 2024, so far we have tested 24 samples, and we could not pick up any metapneumovirus cases. So that means there is no surge of cases in so far in our place. We need to keep a watch. Now, these were the major clinical features of the samples that we got. The major symptoms were including cough and coryza, and the breathlessness, headache, mechanical ventilation was required, and altered sensorium in few patients, and vomiting. Then the age groups also I have mentioned here. So we could get patients in the pediatric group as well as elderly patients. So this is what we have done so far with respect to this metapneumovirus. And as such, it was not a major pathogen that we were focusing so far, but it was there in our panel, so we could pick up these infections. So thank you so much.
Thank you, Dr. Srikumar, for the excellent presentation. And you have been talking about the metapneumovirus human as well as avian, then the potential for mutation, and the thing is that the virus was there in India, and there were a lot of scientific data about the metapneumovirus since 2004. And so there is a clear evidence that this is not a new virus, and that is already existing in India also. So also about the difference between the SARS-CoV-2 and other viruses and the metapneumoviruses. There has been a lot of knowledge has been transferred here. Thank you very much for the excellent presentation. I think now we'll move on to the question answers session. There are many questions on the chat box, and some of them have been answered by Aravind. I would request Dr. Senior, Associate Professor, School of Public Health, to proceed with the question answer sessions. handle the question and answer session. Please.
Thank you very much, Suma Madam. So we move on to the questions. I think some of the questions have already been answered. The question from Amar Patel sir is for the mass medical community. This is going to be a very important session. Okay, so that was just Harikumar sir's question is, how did we conclude that it was there for the past 50 years and only newly detected? May I pass this question to Sharada Madam? Sharada Madam, the question is, how did we conclude that it was there for the past 50 years and only newly detected?
It was evidence is there, serological evidence, even though it was detected in 2001, retrospective analysis of the samples showed the detection of this virus. So it is there. There is evidence that the virus is there.
Probably, sir, would you like to ask the question? So, are you concerned about the 50 years in particular?
Yeah, how did you arrive at 50 years? Because the virus is newly detected and
It's not newly detected.
It is not really.
no,
It is
2001. It
One.
was, yeah.
But
Yeah,
at
it
that
was there.
time, they did research, they did retrospective analysis of the samples was done, and antibody serological evidence was there. That is why we are saying this virus is not a newly discovered virus.
Your preserved
Dr.
samples were preserved
yeah.
samples were detected.
Yes.
Yeah. Okay, right.
Sir, in this study, actually, the oldest sample was only 50 years. That is why, we say 50 years.
Okay, right. Probably it is much earlier it was there, but it was not studied.
We don't have any stored samples.
Yeah.
Okay. So the next question is from Dr. Nasser. Actually, Dr. Arvind has answered it. So I'll put the question once again for the interest of the larger audience to Dr. Arvind. What are the unique challenges and concerns related to HMPV infection in vulnerable populations such as young children, older adults, and those with underlying health conditions? As no specific treatment is available, even giving antivirals doesn't work, as said by Sir before. Is it just symptomatic, or is there any specific treatment guidelines to be followed for the complications if it happens?
Actually, when we are looking at the pathophysiology of metapneumovirus, what it leads to is increase in mucus secretion and then epithelial hyperplasia, airway hyperresponsiveness leading to bronchiolitis and the wheezing episode, airway obstruction. So the supportive treatment is actually aimed at alleviating all these things in the children. And that is usually enough because the pneumonia per se is not there. But in immunocompromised people, elderly people and all those people, and especially in immunocompromised, the major modality of treatment is modulating the immunosuppression. So that is what I showed in hematopoietic stem cell transplant as well as in lung transplant. So what we can do is actually bring down the immunosuppression instead of actually relying on antivirus, because antiviral with pan RNA virus activity, ribavirin, remdesivir, etc., we don't have any clinical data in this metapneumovirus infection. Only the in vitro data is there. And what I believe from that study is that actually we need to modulate the immunosuppression that we are actually using. The second aspect is that majority of the studies actually show that there is around 30 to 40% co-infection with other viruses and other bacteria as well, including pneumococcus. And if the patient is not responding, we need to look at a treatable entity. Suppose there is a bacterial co-infection, probably a targeted antibiotic therapy will be required. So basically, we need not worry that much. Symptomatic treatment is also definitive treatment based on the pathophysiology.
Thank you very much, Dr. Aravind. And the question from, it's not a question from Shaji sir, so was just warning us that the flu season is here and that we need to take precautions. The next question from Dr. Midi Sridharan, which drug is effective in macrolide resistant mycoplasma? I'll put that question again to Dr. Aravind.
We will say azithromycin or macrolide resistant mycoplasma pneumonia if there is no effervescence or symptomatic improvement in 72 hours. In that situation, we have to opt for doxycycline or quinolone. So the first option will be definitely be doxycycline, and usually they respond to doxycycline.
Can you just take up the next question also? Why there is a high rate of mortality? So due to this infection.
Actually, WHO statement came yesterday. There is no increase in mortality due to metapneumovirus infections. Yeah.
Okay. Again, a question from Harikumar Naik, sir. Can avian metapneumovirus infect us? Or HMPV, AMPV, we're talking about the origins. Our daughter and mother or both are the same. So can I ask this question to Sri Kumar, sir?
As of now, we don't have any evidence or any probability that the avian metapneumovirus can infect us, because the receptor binding domains are significantly different. And so we don't expect that they are able to infect humans. So possibilities are very minimal. They have diverged significantly.
Another question from Hari sir is that throughout the presentation, we have used small h MPV and not capital H. And do we have any specific reason? May I ask this question to Sharada, madam?
No, Aravind has given the answer, correct.
Okay.
Yeah,
Madam, I'm just putting it for the sake of the larger audience who don't really look into the chat box.
What Aravind has explained, CDC has given it as a capital H. There is nothing up to date is given as H small letter. And in our virology textbook also it is given as capital H. I also had some confusion whether it is to use small h or capital H, but I saw in all the textbook of virology, it was given as H capital. There is nothing much difference between capital and small letter, Hari.
Okay, so we can use both, madam.
Yes.
Is that the answer? And Suma madam was trying to say or ask whether it is a non-human PV.
Yeah.
Okay, so I think we will. And Amar sir has a question at the raised hand. Sir, would you like to ask?
Yeah, thank you for the opportunity. Any predictions about what viruses are likely to pop up into predominance over the next 5 years? I mean the respiratory ones, going by present trends.
Who is going to take that?
Sri Kumar will take it.
No, there is a question also, like of the 4K samples tested last year, can you give a breakup of viruses identified or isolated? So maybe I can combine both. So
Yeah.
during our testing there is some seasonality. We are yet to analyze that, but we get almost all the pathogens in that panel. So we get Boca viruses, we get SARS-CoV-2 once in a while, influenza A and B. Sometimes it will be streptococcus and mycoplasma, then parainfluenza once in a while, then metapneumo. So we need to do a systematic analysis, but we could pick up all these pathogens in that panel. So that means.
Can I ask one more question, sir, from Dr.
Sure.
Shija? That is how to send specimen to Virology Institute from suspected cases. Maybe that's
I think there are certain couriers which collect these samples and bring to us, and many times even by the patient bystanders also bring the samples to us. So we collect it directly. We don't take samples there, but they have to bring the samples in.
Dr. Shaija, I hope the question is answered. There is a question from Dr. Gauri S. Maybe Aravind can take it up. Any use of vitamin C and zinc in prevention?
Actually, even during COVID time, we have enough literature showing that the vitamin C and zinc did not prevent any infection. Instead, we should be focusing more on our NPI, non-pharmacological interventions like, say, masking, hand hygiene for the vulnerable population.
Okay, so I think that is the end of the questions there. So we have come to the end of this. Before I go on to the vote of thanks and conclusion, I'd just like to ask if Mohan sir is already there, if he wants to put something, or Dr. Rajmohan sir, if you both want to say something.
I would like to know whether Mohan sir has decided to wear mask or not.
Thank you.
I strongly advise, because flu is very much around.
Yeah. Any comments, sir?
Agree. Wearing mask is good, not because something happened in China. Thank you.
Yes.
So we are on time and we are finishing. Rajmohan sir, do you want to say something, sir?
No, senior, you can
Okay.
few sentence of conclusions you can say.
Okay.
Take home message.
So
then you can wind up. Thanks.
Okay, so take home message, I think I would put it as a few M's regarding, it's about metapneumovirus. So a few M's that we need to take home is that it's relatively mild compared to the other respiratory viruses, that we need not be scared about it. The second M is that related to the mutation. We hear, and all the speakers have said that there is a low genetic variability. Another M is on the mortality. It's less than or around 1%. So that's again something we need not be worried about. Another M is that the focus should be on the me or the host factors, because that's where we need to work on, especially those who are immunocompromised and taking steroids and the transplantations and all those things. And another M is on the masks with Mohanan sir's name also starts with M. So starting on asking us on the mask, and we now know what stand to take on the use of masks. And last but not the least, how we should be tackling the media with all this awareness. I think all of us are well placed now to be talking to the media that there is no need of a scare. It's a relatively mild disease and there's no change in the clinical presentations, and it's something that we can manage. Okay, so that is to sum up. I now move on to the vote of thanks. First, I'm indeed privileged to give the vote of thanks on behalf of the organizing team for today's insightful and impactful workshop. First and foremost, I place on record our heartfelt gratitude to our leader, Mohanan Pudumal sir. Actually, it is his thought and vision, support and guidance that made this workshop possible, a reality within a very short span of, in fact, 2 days. Your leadership will continue to inspire us, sir. And thank you for being with us all through the session. Next, I'd like to take this opportunity to thank our professor and head at the School of Public Health, Dr. Rajmohan sir, who took it up from Mohanan sir and very actively called upon the speakers and arranged everything. Thank you very much, sir, for all the things that you did in relation to the workshop. We are deeply grateful to our esteemed international speaker, Dr. Shyam Kotlil, in spite of his limitations with time and weather and all. He has given us a very rich global perspective, and his expertise and insights have truly broadened our insights as well. Heartfelt thanks to Dr. Arvind, who's always with us for all the activities of the School of Public Health. Your talk on the clinical features and management has been really illuminating, and that was very clear, and we are thankful to you, Arvind, again, and a sincere thanks to Sharada Madam. Sharada Madam, we really enjoyed your talk, just like the previous, all through the years in medical college. Your expertise and your talk has been really given with great precision. Thank you very much, ma'am. We extend our warmest thanks to Dr. Srikumar. Your work in the field of diagnostics have really given us the evidence to strongly believe that we can, that it is already there and that we can very well tackle the disease, and a lot of insights into the diagnostics and what we can do. Okay, thank you, Srikumar sir. Thanks to a very special thanks to Suma Madam, who has been moderating the session brilliantly, and she has been steering the program, the discussions right from the beginning. And we are actually lucky to have her at the School of Public Health. So much of vast experience with research and clinics. Thank you very much, Madam. I also express my gratitude to all the participants. There were a lot of dignitaries amongst our participants. I'm not taking names for want of time. And thank you for your enthusiastic presence. In fact, at the time, I think we had around 260 plus participants. And even now, I think there are around 100 of them. Thank you very much. We could not have spelled success without you. And I'll also take this opportunity to thank all those who helped to disseminate the workshop and helped so that there were so many participants here. And for all the organizing team from the School of Public Health, there was Dr. Jisha, there is Atul, there is Sindhu, and there Jalinchechi, and all the technical support from the IT division of the KUHS, Harilal sir and Priya and the team, and all the for all the communications And a special thanks to our academic dean for preparing the brochure for us in very short notice and very well. So let's carry forward the knowledge that has been shared today to strengthen our preparedness. And I'll wind up with a session that will continue this series, perhaps with, we have a food for thought for another M, and that is the mycoplasma. Thank you once again for all your support, guidance, and being there with us throughout the program. Have a great evening and good night. And I hope the metapneumovirus doesn't come to your dreams. Thank you.
Thank you, Senior, for your very informative and crisp Thanksgiving. Now, I once again thank our Binoj also in academy for all the support, because he very silently works for all the support for these programs. Thank you all. We'll call the day. Okay.
Rajmohan sir പറഞ്ഞു കേൾക്കുന്നില്ലെന്ന് തോന്നുന്നു.
കേട്ടില്ലേ? അയ്യോ.
ഞാൻ കേട്ടു.
സാറിന്റെ ശബ്ദം കുറവെന്നുള്ളതേ ഉള്ളൂ, കുഴപ്പമില്ല.
ഇല്ല, ക്ലിയർ ആയിട്ട് കേട്ടു.
ഞാൻ കേട്ടു.
ഞാന് ബിനോജിനോട് താങ്ക്സ് പറഞ്ഞു, നമ്മുടെ ഡീൻ അക്കാഡമി. പുള്ളി അവിടെ സൈലന്റ് ആയിട്ടിരുന്ന് എല്ലാ സഹായവും ചെയ്യുന്നതുകൊണ്ടാണ് നമ്മൾ ഇങ്ങനെയൊക്കെ ചെയ്യുന്നത്. ആക്ച്വലി വെരി reputed and experienced orthopedician sitting in the headquarters, university headquarters, and helping all others to do all these activities. So a special thanks to Dr. Binoj, Dean Academy. Okay, thank you all. So, madam, നിർത്തിക്കൂടെ?
Yes, sir. Thank you.
എല്ലാത്തിനും കാരണം
We can close. എല്ലാത്തിനും
കാരണം സുമമാഡത്തിന്റെ ഐശ്വര്യമൊന്നും. കാരണം വലക്കാലത്ത് വെച്ചിട്ടുള്ള ആദ്യത്തെ നമ്മുടെ വെബിനാറാണ് ഇത്രയും മാക്സിമം അറ്റൻഡൻസ് ഉണ്ടായത് 2 ദിവസം കൊണ്ട്.
Thank you. ശരി, we'll close it.